Okay. Sorry. I'm recording this Zoom. I don't know how well that's actually going to work out. Told me that apparently I'm not good enough for the extended Zoom meetings, and it only gives me 40 minutes. So every now and then, if we go that long, I'll go over here and restart the Zoom so I can do multiple recordings, and then I'll send it out in an email for those people who missed it. There were a few people that reached out so that they wanted it recorded, but that's just what I'll be doing. So I don't actually know what's going to be on the exam. I don't know how long the exam is going to be, how long it'll take you. He doesn't give us that because he wants it to be fair to you guys on which exam review you go to. He doesn't want Mike to go over some things that'll be on the test and me not go over some things. So he just tells us, go over the slides, go over the important things, and those will be on the test, and you'll be blind and you won't be blamed for anything that. So last year, I can go off this. Last year, the exam was 93 questions. It was all multiple choice and true and false, and it took the average person about an hour. I expect that to be the same case. It might be a few less questions because we covered less stuff in chapter 9. That's just a guess, but it should take you about an hour, a little bit over. I would go ahead and plan on like an hour and a half to 2 hours because my neuro exam told me that it was supposed to take an hour and it took me like an hour and a half, and that sucks because I missed something. But yeah, so that's just the basic format. If you have any questions as I go over this stuff, I'm just going to go over the main things. Just feel free to shut me up and we'll talk about it. Do you have a question already?
I think there are some people in the waiting room, the
Oh,
Zoom
really?
waiting room.
Oh, that's awesome. Thank you. Okay. Complicated.
Disable that though, like the waiting room if you don't want to have to like check to admit people. I think
Oh, awesome. Cool. That helps. So for anyone who just joined the Zoom, can you like put like a thumbs up or something if you can hear me in the chat or something? I don't know. Yes, awesome. That's cool. All right. So I explained it to the class, but about every 40 minutes, I'm going to shut the Zoom down and then I'm going to restart the Zoom because it only gives me 40 minutes. So when I shut the Zoom down, just join the same link and we'll pick up right where we left off. This is also being recorded, so I'll post it or I'll email it to you guys when I get it recorded. Okay. So let's just go ahead. I went over the parameters of the exam. It's about 90 questions. This is last year's format. It should be about the same. Multiple choice, true, false should take you about an hour, but go ahead and plan on it taking an hour and a half. That way you have time to make it to where you're going after. Okay, so let's just go ahead and start with chapter 1. Chapter 1 is some pretty basic introduction stuff. And honestly, I think it should be like common knowledge. If it's not, feel free to email me with stuff that you're concerned about that I don't go over. I'll be happy to answer that. But the biggest thing is homeostasis. And so if you have a grasp on that, if you're able to write down everything you know about homeostasis and you have a pretty good grasp on that, you should be good for chapter 1 and the questions pertaining to chapter 1. And it's basically just internal regulation. Homeostasis isn't equilibrium. Okay, it's different. It's maintaining a state of dynamic changes based on what the external environment gives you. So if I get really hot when I'm playing basketball, then obviously that is going to be an input signal. Okay, my body temperature is going to rise, and it's going to integrate. It's going to go to my central nervous system, and then I'm able to create an output signal for my an output signal goes efferent to my sweat glands, and that produces a response to get me to start sweating, and then I'm able to cool off. That's a basic example of homeostasis. They're oftentimes accomplished from positive and negative feedback loops. So any failure to compensate for environmental response or what is it? What's the word? Stimulation, I guess, situations would be, disease and death. Oftentimes, most cases, disease, unless you're exposed to really bad stuff. In that case, it'd be death. But like I said, it's mostly feedback loops that control this. And they're categorized in a positive and negative, but they're also categorized into reflex, which is extrinsic control, and local or intrinsic feedback loops. I guess I would probably know the difference between these two if I were you guys. That reflex uses the nervous system or endocrine system, and it's body-wide. It's more like widespread, whereas local happens within relevant organs or relevant body compartments, and that has a lot to do with metabolic activity. Okay. Yeah, I'd say most of the time when you have like environmental factors like heat or temperature or exposed to some kind of panic situation, your sympathetic response revs up. Those would mostly be reflex or extrinsic controls. And then stuff that's local or intrinsic would be like your membrane potentials or the pH or the salinity of certain solutes or solutions in your body. Stuff like that would be more local control or intrinsic. Okay, so we'll go into a few more examples of that. Like I said, if you guys have any questions, just let me know and I'll do my best to answer them. Negative feedback loops, like I said, like is temperature. If you get cold, the input is cold. And so the negative feedback loop is the opposite of the external environment. So if I get cold, I'm going to feedback by trying to make myself hot. And that's what it means to be negative. It's going to be the opposite of the stimulation. Okay. You also have that in blood sugar regulation, glycogen breakdown, glucose breakdown, stuff like that. Whenever you have glucose enters the bloodstream, and insulin gets secreted and you break down that glucose, that's another example of negative feedback. Or homeostasis. Osmoregulation, whenever you get dehydrated, your body secretes ADH, which is antidiuretic hormone, and so you pee less, you retain more water, and that way you're able to survive a lot longer than actually just not maintaining that homeostasis and giving up and dying. Okay. Some positive feedback loops, one of the biggest ones is the process of giving birth. That is pretty interesting. It's a pretty good example of how physical stimulation can activate certain neural pathways. And so whenever the baby lowers down from towards the cervix, that mechanical stimulation on the cervix releases, stimulates the brain, and the pituitary gland releases oxytocin. The oxytocin gets into the bloodstream, gets to the uterus, dilates the smooth muscles around the uterus, able to open up and push the baby out. And that whole thing goes in a loop as the baby's going down into the birth canal. So that's an example of a positive feedback loop. This, stimulation is the baby's getting closer to being born, so let's help it actually get born. So that's positive. Opposite would be negative. Okay. So that's basically it for chapter 1. Does anybody have any questions about homeostasis? Okay. It is pretty straightforward. The test questions on homeostasis should be pretty easy. Basically just know positive and negative feedback loops and why homeostasis is important. Okay, so chapters 2 and 5 get a little bit more complicated. This is usually where people struggle the most. In the beginning of physiology classes is definitely where I struggle the most. Hopefully I'm able to explain it. But the first part, we will be talking about membrane dynamics and transport processes. This is the foundation of all physiological phenomenon in the body, would be this. So this is why it's so important. And as a trade-off, that's probably why it's difficult to understand. Okay, so you have simple diffusion. That's the easiest one. Molecules pass freely just based on concentration gradients. And then you have factors that influence that simple diffusion. And I would know those factors if I were you. The biggest ones are definitely lipid solubility and molecular size. Okay. You also have concentration gradient. If you don't have a very big gradient set up, then there's not going to be very rapid diffusion. That diffusion is going to be very slow and very random and hard to predict. If you do have a very established concentration gradient, then you're able to predict where it's going to go, how much force that diffusion is going to exert, and just stuff like that. And you also have surface area, which is a little bit lower of an influence on diffusion and composition of the lipid layer. It's usually pretty about the same. So that one can be pretty consistent across different compartments of the body. Yeah, so lipid solubility, obviously non-polar things are going to diffuse into the cell membrane a lot easier than polar things. And that makes sense because the phospholipid bilayer is made out of lipids, and therefore it is non-polar on the outside, or non-polar. Tails of the phospholipids aren't soluble to water because water is charged, water is polar. That's pretty fundamental to know, so I would definitely know that. Cholesterol is a type of lipid, and cholesterol actually helps regulate membrane fluidity. So I would understand that cholesterol helps with that fluid mosaic and the phospholipid bilayer. And there's two types: high density lipoprotein, which is good; low density lipid protein, which is bad. I don't know how much that will be on the test. If anything related to cholesterol, I would just know these two things. That would be it for that. Okay. All right. So facilitated diffusion often gets confused a lot of times because it's not exactly diffusion, but it is very similar. The only difference is that something actually has to start it, actually has to allow it to diffuse. So the force for a concentration of certain solutes to go across an area or to diffuse across an area is there, but it's being blocked by something. And whether that's being blocked from an ion channel or a carrier hasn't opened yet, whatever the case may be, the diffusion is blocked. And so to be able to get that diffusion that wants to diffuse, you have to facilitate that, or you have to open ion channels or open those carriers. Okay, and that's often done with water, ions, stuff like that. Those are accomplished with leak channels or gated channels, and those can be gated by ligands, voltage, mechanically, whatever the case may be. Those just allow ions and molecules to diffuse across certain areas because they want to, but it has to be facilitated. It doesn't necessarily require ATP. Something just has to start it, and then it's kind of a cascade and it goes on its own. Yeah, so that's channels. And then carrier proteins are a little bit different. If you remember Dr. Edwards' analogy about canals, that's a pretty good analogy. I like that one where it opens and closes. Did he do that one, or did Dr. Sudwigs do that one? Yeah, he did canals. Great. Okay, awesome. Yeah, so it's a lot like a canal that opens on one side, the extracellular side, and something comes in because it has high affinity, and then it closes and it opens on the intracellular side and releases it because it has low affinity, and then something else binds because it has high affinity. Yeah, so that's the basic principle for carrier proteins. And all of these are subject to competition. That's where you get a lot of drug-drug interactions and what drugs are effective. Competition is based on affinity for these carrier proteins or for these channels. And then saturation. There comes a point where only so much ion or molecule or whatever the case may be can go in at one time because you only have so many channels or you only have so many carriers. So that's the rate limiting step, is saturation and the amount of ion channels. If you have a lot of ion channels and a very low saturation of molecules, then you won't have a problem with your rate limiting step. But if you have a high amount of saturation and a low amount of receptors or channels, sorry, then you're going to have, you know, low permeability. Okay. Any questions about that? Cool. All right. Active transports. So this actually, on the other hand, requires ATP, and it comes in a variety of forms. You can have primary active transporters are often called ATPases. That's a pretty good indicator that what you're talking about is an active transporter. Examples of that would be the sodium potassium pump, where 3 sodium go out of the cell and 2 potassium go into the cell. If I know Dr. Edwards and how much he likes neurobiology and setting up concentration gradients, that will definitely probably be on the test. Like how many ions go out, what ions go out, how many ions go in, what ions go in. So I would know that one for sure. And if it's not on the test, the worst case scenario, you just know it for the rest of your life, and that's great. That will definitely come up later on. Secondary active transporters don't use ATP. Okay, so primary active transporters do, secondary active transporters don't. But they still rely on energy. But the difference is, what kind of energy are they relying on? They're not relying on ATP. They're relying on energy that is that comes from ions passing through their concentration gradient. So an example of that would be the SGLT, or the sodium glucose transporter. It's over here on the right. It's over here on the right. Normally, sodium would not go through this. Okay, sodium would stay on the outside. But since glucose, it's a penetrating solute, since it can go through this carrier, sodium can't, but glucose can. It's going down its concentration gradient. Sodium just hitches a ride on that and takes advantage of the energy that's created to move glucose down its concentration gradient for sodium to tag along with it. That's called a symporter. That would be an example of secondary active transport. Okay, so the sodium is using the energy that glucose is able to generate by going down its concentration gradient to just hitch ride and follow into the cell. Okay, does that make sense? Okay. Awesome. If it doesn't, feel free to ask me. I'm sure if it doesn't for you, then it doesn't for a lot of other people, and that would be cool. I'm sure they would appreciate it. Okay, so that's pretty much that. Now we'll go into something that I actually struggled with for a really long time, and that's the difference between osmolarity and tonicity. Okay, so if you struggle with this one, don't feel bad. That's perfectly normal, and that happens to a lot of people. Okay, so osmolarity is just the molarity of something times the amount of solutes per molecule. Okay, so let's say we have, let's see, we'll make it simple. We have a 1 molar solution of glucose and a 1 molar solution of magnesium chloride. Okay. So glucose doesn't dissociate. It doesn't ionize. Okay, it stays the same. All right. And to calculate osmolarity, it's basically, like I said, molarity times the number of solutes per molecule. And so in this case, it's 1 times 1. Intuitively equals 1. And then for magnesium chloride, the story is a little bit different. Okay, or calcium chloride, the same thing, or sodium chloride, it's a little bit different because the number of solutes changes, because this actually dissociates into magnesium and then 2 ions of chloride. Okay, and so you times that by how many? Awesome, right. And so then you get 3. And so instead of the molarity is 1 for each of these, but the osmolarity is 1 osmol for glucose, right? And then 3 osmol. Can you guys see that? 1 osmol for magnesium chloride. Okay, cool. That's going to be a little bit important later when we get into hypotonic versus hypoosmotic. Okay, and then percent solution is the easiest part of this. It's basically just like what's 5 divided by 100, 0.005, then you just do the decimal place and you got your percentage. So if you have 5 grams of sucrose and 100 milliliters of solution, then the percent solution is going to be 5%. So this is where it gets a little bit more complicated. There's 2 types of solutes, like I said. You have penetrating, which is like glucose or urea. So I'd know those 2 if I were you. And then you have non-penetrating, which would be your sucrose or your salt, because these move in and out. Or no, sorry, non-penetrating doesn't move in and out, okay, because they're charged. Usually they have polar charges, which doesn't allow them to pass through the lipid bilayer immediately, okay? And those set up the concentration gradients, whereas the penetrating ones can enter the cell. Glucose and urea can enter the cell as freely as they want to, just depending on their concentration gradients, and therefore they don't really matter too much in tonicity. But osmolarity, on the other hand, accounts for both. Okay, like I said, it matters the number of solutes. Okay, it doesn't matter the type, it matters the number. All right. Whereas tonicity only accounts for the non-penetrating solutes. Okay, so that leads into comparing osmolarities. This one wasn't on Dr. Edwards' slides, so if this looks new to you, that's for a reason. So let's compare really quick. Let's say you have a solution that is 1 osmol of glucose, and then skip that one, and then 1 osmol of sodium chloride. Okay, in a tonic solution, those would be the same. Okay, but this is an osmolarity problem, and so solution A is actually, well, I'm sorry, osmols does something different. Sorry, that doesn't matter if it's non-penetrating or penetrating. So this right here, what is that? Is that penetrating or non-penetrating? Penetrating, right? These two right here, is that non-penetrating or penetrating? Penetrating, great. Okay, so just, it doesn't matter if it's non-penetrating or penetrating. What you're looking at is the number for osmolarity. Okay, and so A is isoosmotic to C, because it has the same amount of osmolarity. Okay, but if you have 1 osmol of glucose and 2 osmols of glucose in solution B, then A is going to be hypoosmotic to B, and on the reverse, B is hyperosmotic to A. And that's just based on osmotic pressure. Which one has the most osmotic pressure? B has the more osmotic pressure compared to A, so it's hyperosmotic. A has less osmotic pressure to B, so it's hypoosmotic. Okay, but this, I don't expect that to be as drilled as tonicity, but it is good to know the difference. This one, there probably will be questions on the test about this one for sure. This one's a little bit different. Now we're looking at what? What are we looking at? These non-penetrating or penetrating solutes? Non-penetrating. Okay. So the non-penetrating ones are going to stay in the cell. Okay. And so that creates a difference between the inside and the outside. Okay. So step number 1 is to look at the cell and the solution and to consider them separate compartments. And then step number 2 is to look at the amount of non-penetrating solutes inside the cell relative to the outside. Okay. And then we look at how much is in versus how much is out and determine what the solution is based on those 2 concentrations. And so if you have a cell that has a bunch of solutes inside and the solution doesn't have hardly any, then what's going to happen to water? It's going to go into the cell, because that's just simple osmosis. And when that happens, you get a hypotonic solution compared to the cell expands. Isotonic means it's the same, stays the same. Hypertonic meaning that there are a lot more solutes outside than inside, and the water goes out and the cells shrink. Okay. And this is always relative to the solution. We're always talking about the solution. Okay. So the solution is hypotonic. Okay, not the cell. Okay, that gets a little bit confusing for me sometimes. All right, any questions about osmosis and diffusion and stuff like that? Okay. All right, so membrane potential. This one is also a big one. Resting membrane potential, the difference in charge across the membrane is essentially all that is. And that's measured in voltage, usually millivolts, and that's created mainly by the sodium-potassium pump. And it depends on the concentration and the permeability of key ions. So this permeability right here, that gets important in the Hodgkin's equation, the Goldman equation, whatever you call it, I can't remember. But each ion is a little bit different based on the concentrations in and out of the cell. Sodium, obviously, the most of it is on the outside. Potassium, most of it is on the inside. Okay, and that's what sets up that concentration gradient. That's what sets up the membrane potential, or the resting membrane potential. Okay, and then you have chloride, which is the anion in most cases, and it's also a little bit different. Okay, and so let's talk about equilibrium or reversal potential. This is, I can't remember how he said it, but Dr. Edwards was talking about if you're like asleep, then you might want to wake up for this. So this one's probably pretty important, I would say, if you were there for that lecture. And this is usually where it gets the most complicated for people to wrap their heads around it and what's actually happening here. But for every ion, the reversal potential is different. And that's because every ion has a different concentration inside of the cell versus outside of the cell. And that creates different forces chemically and different forces electrically to drive the ion in or out of the cell. Okay, so I'm trying to think of the best way to explain this. Did it make sense to anyone, like how Dr. Edwards explained it? Did that make sense at all? Because I'm trying to decide if I should explain it a different way or if I should try to explain it the way he did. I'll just reiterate what he said. So let's say you have a membrane. This is inside. Actually, let me just do that right here. That's inside. That's outside. So for potassium, is it the most concentrated? I just spit everywhere. Is it the most concentrated on the inside or on the outside? Okay. So I will do it the same way he did. The size of the symbol is the amount of solute in the cell. Okay. So if the channel is open, okay, let's call it a leak channel or whatever. If the channel is open, based on the amount of concentration inside versus outside, it makes sense that that driving force is going to cause potassium to leave. Okay, so potassium leaves, and now you have more potassium on the outside and less on the inside, but you also have something else that happens. Okay, you also have negative charge left behind. Right, because potassium leaves, it brings its positive charge with it, becomes more positive on the outside, less positive on the inside, and now you have another factor. This is the chemical factor, the concentration of the solute itself, and the electric factor is the amount of charge left behind, or the amount of charge it leaves. Okay, and so as this leaves, the cell becomes more negative, right? And this becomes smaller. This becomes bigger, more positive charge. So there comes a point where it doesn't make any sense for potassium to leave the cell because you have these two forces keeping it inside. And to quantify that, we need a number. It needs to make sense. And that number happens to be negative 90 millivolts. So when there's enough negative charge on the inside of the cell for it to reach negative 90 millivolts compared to the outside, then there's not enough driving force for the potassium to leave the cell because it doesn't want to anymore. Okay, if it was negative 80, if it was less negative, potassium would still leave the cell. But the amount of membrane potential that keeps sodium in the cell is negative 90 millivolts. Does that make sense? Okay, great. And the same goes for sodium, just reverse. Okay, so now, sorry, that might bother people if I use my hand. So now it's different. Where is sodium most concentrated, inside or outside? Awesome. So since sodium is more concentrated on the inside, less concentrated, or more concentrated on the outside, less concentrated on the inside, what force is driving sodium into the cell? Electrical or chemical?
Chemical.
Right. So chemical goes in, this becomes more positive, this becomes more negative. Concentration of sodium gets higher, concentration of sodium gets lower, right? And now, once I reach what value? 60. Plus 60. Once I reach plus 60, no more sodium is going to want to come in because it doesn't make any sense. It doesn't want to anymore because it doesn't have the chemical driving force and the electrical driving force, the membrane potential, is keeping it outside. Okay. Feel like that was pretty decent. Great. Awesome. All right. So now let's go over communication. And this stuff is important, what I just went over, because cellular communication would not be possible without those chemical gradients. And it's not just random. All this stuff that we cover in the beginning stages are in every single concept we study after that. Okay. All right. Okay. So physiological signals come in a variety of different types, obviously, like everything else. And those are either electrical or chemical. And you get different actions based on what type you use. So changes in membrane potential would be examples of electrical signals, and then neurotransmitters or hormones secreted by cells. Those would be examples of chemical signals. And these are all responsible for communication in the body. Target cells respond differently to each signal type. And then, that's great. Give me a little reminder.
I think you're good to keep going.
Yeah. Oh, awesome. To me. That's great. Cool. So I wonder how many times I'll do that. So let's go. Where was I? Okay, we'll just start with short distance communication is often done by gap junctions. If you remember me talking about gap junctions, those are often done electrically because it's easier to send electrical signals through gap junctions than it is chemical signals. So those are almost always electrical. You have like contact dependent things that would be like autocrine signaling. Paracrine signaling is adjacent to. So auto means self. So it makes sense that autocrine signaling would be stuff for cells secreting a ligand, so it actually does it itself. And I'm trying to think. Someone asked a professor that I know an example of autocrine signaling, and he gave one that was pretty good, but I can't remember. So apparently it wasn't too good. But I can't remember. I don't know. It happens, I'm sure. And the paracrine signaling is most of the endocrine and neurocrine signaling in the body, and it just sends out chemical neurotransmitters or transmitters to affect the neighboring cells. And that's where most of the signaling comes from, especially in endocrine signaling. And then you also have long distance communication, which would be done by bulk flow. Does anyone know what bulk flow is? Bulk flow is basically just the blood or the lymph. Okay. And that's where your long distance communication comes in, because the blood circulates all throughout the body and so does the lymph. So if I have a hormone that I want to secrete into my entire body, it makes sense. I'll just go ahead and put it in the bloodstream and then it'll go everywhere. Okay. And that's where most of the long distance communication comes from, and hormones are secreted like that. Okay. You can also get that in the nervous system too, since the nervous system runs everywhere. So you can get neurotransmitters that are done long distance. Okay. Any questions with that? Cool. Did you have one?
Yeah, just for paracrine signals and autocrine signals, if a signal both acts on a receptor on its own and on others, we consider paracrine.
So if it, that's a good question. I don't know if I should restate the question. The question was, so that the Zoom can hear it. The question was, if a cell emits a receptor and it binds to its neighboring cells and the cell itself, is it considered paracrine or autocrine? Is that your question? Okay, I would say it's both. Okay, so if it emits a neurotransmitter and it binds to itself, then that's autocrine. If it binds to other cells, then that's paracrine. So I'm sure there's a lot of examples where the cell does secrete a lot of different signals and it does bind to itself. And that would be an example of positive feedback loop. So that's a good question. All right. Okay. So with signaling, chemical signals are the most diverse, and they come in a variety of different pathways. And the pathways we go over, I would definitely know those for the test. I think he actually stated in the lecture that he wanted you to know those. So I would. Okay. And it's all the same pretty much. It's the same phenomenon. You get the signal molecule, binds to the receptor or the transducer, whatever you want to call it, and it activates intracellular signaling molecules that then cascade and go to different other effectors and target proteins, and that elicits a response. Okay, you get a lot of different types of responses for different types of second messengers, and then you get a lot of different second messengers for different types of first messengers. So this is one of the most diverse things in science. And if you're ever like, I can't remember exactly where it is, but there's a hallway on the 3rd floor of LSB near one of the labs where they have a huge poster of all the different signaling pathways in the human body, and it's just ridiculous. I have no idea who would ever want to map that out or read that, but it's there and it's pretty cool to look at a glance. Definitely wouldn't recommend doing any more than that unless you want to learn something. It's okay. So let's go ahead and go into the different receptor types and signaling types. You have G protein coupled receptors. This is responsible for the majority of pathways in the body. And that's because it's so diverse. That's because it has many different effects that it can do. And one of the pathways that Dr. Edwards probably wants you to know is the cyclic AMP pathway or the CAMP pathway. And this one's pretty easy relative to others because I was in Dr. Sedgwick's class and he mentioned this. A goes with A. Okay, so you get the molecule, activates the G protein. The effector G protein goes to adenylyl cyclase. Okay, that's its effector target, which then converts ATP, so adenylyl cyclase, ATP, then converts, sorry, that's not right. Yeah, it just activates. Okay, so then it activates cyclic AMP, which then activates protein kinase A. So adenylyl cyclase, ATP, cyclic AMP, protein kinase A. So that one's pretty easy. Pretty cool. And then protein kinase A goes on to phosphorylate a bunch of different other proteins and then get various cellular responses. These can be nuclear responses like transcription factors. They can be other molecules that go to different places. It really just depends. And they probably didn't specify which one for a reason because there's just so many. All right. So that's G protein coupled receptor for the cyclic AMP pathway. You also have G protein coupled receptors for the PLC pathway, which is another one that Dr. Edwards probably wants you to know. This one's a little bit different, a little bit harder to learn, but it's basically the same process. You get the single molecule, activates the G protein coupled receptor. It's called a G protein coupled receptor because the G protein is coupled with the receptor. So the G protein then, once this is activated, goes to its effector protein. In this case, it will be phospholipase C. Phospholipase C has the ability to cleave a membrane phospholipid called PIP2. It's not in here, is it? No, it's not in here. But that I don't think that's important, but it's PIP2. Okay, and then that membrane phospholipid PIP2 is cleaved into two molecules, DAG and IP3. IP3 is a second messenger. That's important. He might ask you that. What is IP3? Is it a second messenger? Yeah, yes. Okay. So what does IP3 do? It then binds to the endoplasmic reticulum and releases stores of calcium. Calcium is also a second messenger. So that's pretty fun. You get second messengers for second messengers. That's great. And then you get cellular responses like muscle contraction is dependent on calcium release. And so the PLC pathway would probably be pretty important in muscle contraction, if I had to guess. And then, so that's one side of it. The hard part about the PLC pathway is that there's two sides of it. The first side is the IP3 side. The second side is the DAG side. And DAG actually binds to it and activates PKC, PKC with help of a protein, then phosphorylation, then induces its own cellular response. Okay, protein kinase C, that's what PKC stands for. So yeah, two sides to that one. That one's a little bit more complicated, but I would definitely memorize that one and know what that does. The PLC pathway is often used for metabolism too. So that might be important to know. Come back. Okay. The arachidonic acid cascade. If you go into Learning Suite and you look up like the study guide exam, this one's listed on the study guide, so I guess this one's pretty important too. But the arachidonic acid pathway is really important for inflammatory responses, and that is done by the enzyme PLA2. So you get this membrane phospholipid binds to PLA2 and then activates and creates arachidonic acid, which is another second messenger. Second messenger is really important in these pathways. Okay, that second messenger then goes on to release prostaglandins or thromboxans or leukotrienes, just whatever the case may be. That one's really important in inflammatory responses. That one's pretty simple too. I would just know PLA2 and arachidonic acid and what the outcome is. right? Calcium signaling. Does anyone have any questions before I keep rambling? Calcium is a second messenger. Yeah. So it's the most versatile second messenger there is. It is used for muscle contraction. It's used for neuroplasticity. It's used for certain metabolic pathways. Bottom line is calcium is extremely important. And it's stored in the endoplasmic reticulum, and it's moved by active transport. And rapid increase of calcium inside the cell triggers certain responses. And that can be a calmodulin or other calcium binding proteins like the snare complex for vesicle transport at the post or the synaptic cleft. It can alter protein activity, whatever the case may be. I would just know that calcium is an extremely important second messenger, and it gets into the cell via the voltage-gated calcium channel. Right? All right. All right, so you also have some other, like, if there were a miscellaneous category for neurotransmitters, these would definitely be in that. And that would be gases and like steroids and stuff. But gases, I would basically just know nitric oxide and carbon monoxide. And nitric oxide is released and produced by endothelial cells and neurons, and it causes vasodilation. It also activates the cyclic GMP pathway. It's a neurotransmitter, neuromodulator, stuff like that. This kind of information would just be like recall, I think, personally. Some multiple choice question about what nitric oxide does, what carbon monoxide does. That would probably be as much as you need to know for these. Okay, carbon monoxide targets smooth muscle and neural tissue. If you want to know what it does in neural tissue, a lot of times it's used for plasticity and initiating that, and initiating like circuitry wiring and stuff like that. Okay, and then smooth muscle, it just dilates it, and it also activates the cyclic GMP pathway. So that's pretty convenient that they both do the same thing with that. All right, that does it for signaling. Any particular questions for signaling?
So would nitric oxide and carbon monoxide be considered other second messengers then?
I would classify them mainly as first messengers, to be honest. But knowing the versatility that the body has and how many second messengers there are, I wouldn't be surprised if it does act as a second messenger sometimes. But if there's a question on the test and it says, which of the following is a second messenger, and you have stuff like nitric oxide, carbon monoxide, I don't know, and then a couple more options, and then you have calcium, the answer is definitely. Okay. All right. Chapter 8 is just an introduction to neurons and synapses. Were there any more questions about signaling? Okay. All right. Let me check. On the Zoom has a question. All right. Okay, so this is neural anatomy, or the neuron's anatomy. And I would definitely know this just because it's good to know. You have the dendrites, the cell body, which is the soma, the nucleus is in there, the apparatus, all your cellular organelles are in the cell soma. And then you have the axon, often myelinated, doesn't have to be, but it's often myelinated. And then you have the axon terminals. And the direction of neuronal signals always, or not always, but most of the time goes from the soma to the axon terminal. So from the dendrites, it receives it, gets into the soma, those graded potentials, goes through the axon into the axon terminals, where it then propagates. Okay, so do you guys remember this being in the lectures at all? I don't either. This was on like the review slides that were provided for me. I'm not even going to really go into this because I feel like that would just give you information that you don't need. But basically, you just have like these microtubules assist in getting nutrients and stuff down the axon into the axon terminals. That's basically all that is. Okay, so there's glial cells, which are really dependent on where they're at. That dictates what they do. So you have the peripheral nervous system, glial cells. Those would be stuff like Schwann cells, and then satellite cells. Satellite cells support the cell bodies, help set up the what's it called? I can't think of the name. The network outside of cells. Okay, Schwann cells form the myelin sheaths and secrete neurotropic factors. If you remember the time where I lectured, I also talked about neural repair. So that would be good to know that you have neural repair in the periphery, but not in the central nervous system. So I would definitely know that too. Okay. Was anybody not there that has a question about that?
Can you say that one more time? If you just said?
Yeah. So in the periphery, you have Schwann cells. Those are the myelinating cells of the periphery. They're able to regenerate neural tissue after damage. They can create these things called regeneration tubes, and basically the axon just runs back through it, kind of regrows itself like a lizard tail, just as long as it's connected to the cell body. But in the central nervous system, you don't have Schwann cells, you have oligodendrocytes. And those actually do the opposite. They inhibit neural repair. Okay, because the spinal cord and the central nervous system is already well developed. There really doesn't need to be a whole lot of changes in circuitry. There can be plasticity, but changes in overall circuitry would cause a lot of problems. And so oligodendrocytes actually secrete these proteins. Does anybody know what those proteins are called? Called no-go proteins. Okay, so those no-go proteins prevent formation of neural repairs. Okay, so that's the difference there. Then you also have epidermal cells that produce the CSF. Do you remember the last lecture? I think we talked about those. Astrocytes are really important in setting up the blood-brain barrier. They're actually like one of the only things that do that. And then microglia are your immune cells of the central nervous system. Okay, so now let's get into the Nernst equation. I don't think this will be. Did he mention that you needed a calculator for the test? Then I don't think this will be very much on the test.
Sorry to interrupt, but he did say like you might need to do more qualitative judgments, like if an ion concentration increases, what would that have?
So that's what I was about to say. I think that will be on the test. What would happen if I change this? What would happen if I change this? Okay, so knowing how that equation works would help you understand what would happen if I do change those. I'm trying to think of a good example to explain that, but basically you're going to get, let's say I change the ion concentration out of the cell. How does the equilibrium potential or the reversal potential change for that ion? And that just depends on how the equation works out. So let's go. Does anyone have a calculator that can do logarithms? Just real quick. Example. Hopefully it's big enough to actually make a difference. But what is it? 125 out. I'm trying to think. This is bad. UTA should know this. Here it is. Okay, let's go ahead and take. Oh, dang it. That's not. Let's go ahead and take the example in the membrane potential slide for, let's say, potassium, since it's one of the biggest ones. And let's go ahead and do outside the cell is 5 micromolar, and then inside is 125. Anybody, well, actually, sorry. Anybody go ahead and put that in 61 times the log of 5 over 125 and tell me what that is. I'm good at math, and I'm definitely not that good at math.
Negative 85?
Maybe 85. Okay. So that's pretty close to what we know is the resting membrane potential for potassium, negative 90, right? But it obviously fluctuates. And so we expect to see a fluctuation just depending on what type of cell it is and how much concentration we have. So let's say we increase the, let's say we increase the amount of concentration of sodium or potassium, I'm sorry, outside the cell. Let's increase that, I don't know, times, and say that's 50. And then let's decrease the bottom of it 10 times and say that's 12 and a half. Okay. Then it would be 61. What do we expect before someone puts that in? What do we expect to happen to the equilibrium potential if the concentration of potassium outside the cell increases and the concentration inside the cell? So I'll go ahead and do this really quick. The concentration inside the cell increases. What do you expect to happen to the equilibrium potential after that? Would it get more negative or would it get more positive? Okay. Does anyone have that value? 36. 36 positive? Positive 30. So that's also a pretty big factor too. If the top is bigger than the bottom for a log, then you're going to get a sign change. If the bottom is bigger than the top, then you're going to get a sign change. And so when we play around with this, you'll get sign changes and you'll get different values. And so that's kind of what we expected. If the potassium on the outside gets a lot bigger, then your equilibrium potential is going to change. All right. Does that make sense? Do you want to have any questions that want me to do it again? Yeah.
So the equilibrium potential is of the inside of the cell. So it'd be like a negative value if there's more potassium on the outside. That makes sense.
Okay. Okay. So I think that covers that. If you have more questions about that, I know that's a really tricky topic, so feel free to email me questions about that, or if you want to talk in person, then I have office hours on Wednesday from 2 to 3. I know office hours on Friday might be cutting it a little bit short. I keep doing that. So I can definitely talk about that with you later on if you want. Okay, this one is a little bit different. I don't think, honestly, it will be tested on this one too much either, because he didn't tell you to bring a calculator. But there's two differences between the Nernst equation and the Goldman equation. And that is, you have to incorporate relative permeability. So you remember that value on the very right-hand side of that slide I just showed you, where it showed you permeability. That's basically just telling you how easily can the ion go in and out of the cell. And so that's important if you want to know the total membrane potential. So on the Nernst equation, we were looking at equilibrium potential or reversal potential of a particular ion. Now we're looking at the entire membrane, the whole cell. So we have to look at each particular ion's concentration inside of the cell and outside of the cell. So that's the biggest change, is you have to add permeability. There's one more change. I don't know if you catch it just by looking at it, but if you have a negative charged ion, it's actually flipped, where the concentration inside is on top and the concentration outside is on bottom. Okay. That would be all that I would really worry about. Yeah.
Would it be flipped for the Nernst equation too?
No. So that's the difference. So the Nernst equation, if it's always out over in, okay, no matter the sign. Z changes because the charge changes. So if we're looking at chloride, you would put negative on this. If we're looking at potassium, that would be positive. So that's a good question. But for this one, it's the concentrations are flipped and there is no Z. Okay. Good question. All right. Any questions about the Nernst equation or the Goldman equation? All right. Graded potentials and action potentials. All right, so I would definitely know the difference between graded potentials and action potentials and what exactly they do and how exactly they do it. That would be my biggest concern for the test. And graded, I like to think about it as graded potentials produce action potentials if certain conditions are met. And that's based on frequency and the amount of stimulation these graded potentials produce. So graded potentials are created in a variety of different ways. They can be created mechanically, chemically, voltage gated, doesn't matter. But those have the most variety. Action potentials, on the other hand, are only created by voltage and voltage gated channels. So changing the membrane potential so much that it causes an action potential. Yeah, so the cell is negative 70 millivolts at rest. Usually the graded potentials change that. And the threshold, I'm sure everyone remembers that, is negative 55. If it gets there, then you get the action potential, right? So that's how I think of it. These three terms, I would definitely know those, okay? Those are pretty important. Depolarizes becomes less negative. Okay, so if anyone's confused about that, I'll just do this really quickly. It helps me remember it when I took neuro in my undergraduate. So you have negative 70 here, and then 0 up here, right? If I'm down here, I'm pretty polar, okay? Because I'm away from 0, I'm charged, right? But if I get closer to 0, if I get to like negative 50, then if I'm there now, I'm less polarized. And so I'm becoming less negative and I'm depolarizing. Does that make sense? Yeah. Okay. On the other hand, if I go down, or let's go even further, if I go down and I'm at like negative 110, right? I'm more negative. And so now I'm hyperpolarized because I'm more polar than I was. Okay. And that's all relative, right? All right. So let's get into graded potentials really quick. They reflect stimulus strength. They're varied and they can be done through multiple different routes. But the stronger it is, the more change in voltage you have, the greater potential you have to reach threshold and cause an action potential. They lose strength as they move through the cell. That's a property that I would definitely be familiar with. And if they're strong enough, this is one of the conditions that I was talking about. If they're strong enough, they'll reach the trigger zone, which is the axon hillock, and they'll be able to initiate an action potential. Right? I would know these two types for sure. These are two types of graded potentials, and I would know how they work and what they're used for. So to explain that, the excitatory postsynaptic potential is used to excite neurons, hence the name. And it works by depolarizing the cell, okay? Bringing it closer to threshold. I feel like this is going to be useful for more concepts, so I'll go ahead and draw it. All right. So I've got resting here, which is negative 70, and I got threshold, which is negative 55. All right. So if I have an excitatory postsynaptic potential, right, this is time. All right. If I have an excitatory postsynaptic potential, I'm depolarizing the cell. So what ions are going in? Sodium. Or what? Sorry. I got confused because you said potassium. But it's sodium, right? Sodium enters the cell, okay? And then it gets more positive, right? Because it's entering the cell, it's bringing its charge with it. And it gets more positive, right? But let's say that it doesn't have enough strength to reach threshold, then it just dies, right? It dissociates within the cell body, okay? Let's keep going. And let's say that we have an inhibitory postsynaptic potential. What's happening here? The opposite. Potassium, which also has a positive charge, is leaving the cell. Or I'd be really impressed if someone could explain this to me. What happens with chloride if you have an inhibitory postsynaptic potential? How? Yeah.
It's going into the cell, but because it's negative, it's also
Awesome.
for polarizing it.
Perfect. So it's basically these two things are really dependent on what ion's going in and what charge it's bringing with it, or what ion's going out and what charge it's taking with it. And so if I have chloride going into the cell, what's going to happen to the membrane potential? It's going to go down. But if I have potassium leaving the cell, it's the same thing. It goes down, right? Because the potassium's leaving the positive charge and the chloride's taking in the negative charge. So it also goes down. Yeah, okay.
Could sodium also leave the cell and cause the same effect?
That's a good question. Just based on the fact that sodium is mainly on the outside of the cell, I would say that chemical gradient is strong enough to keep it from doing that. But if it did, it would definitely do this. Yeah, that's a good question. All right, so that's basically what it is. You have inhibitory postsynaptic potentials, which hyperpolarize the cell, and then you have excitatory postsynaptic potentials that depolarize the cell. Okay. All right. Summation is pretty fun. I like it. And there's two different types. I think we went over it last lecture, and I would definitely know the difference between these two types. You have spatial. That's when they come from different postsynaptic neurons, and they reach the cell body at approximately the same time, and they add together. Let me see if I can go ahead. Here's the nucleus, and go ahead and draw a couple dendrites. So let's say we have synapse there and synapse here. And let's say they're both excitatory, right? So they depolarize this cell. Okay, so spatial would be where these two come in at the same time and they amplify each other because they're here at the same time. All right. So let's say this one is, I don't know, depolarizes the cell by like plus, I don't know, this is really extreme. It doesn't happen at this magnitude, but let's say this one depolarizes the cell at like 15, and this one depolarizes it at a more physiological probable rate. And let's say it does it by 5, okay, 5 millivolts. That one does 15, this one does 5. Let's say the resting membrane potential is -70. All right. So if I go ahead and add these two up, what does that equal? It's plus 20, right? Plus 20 millivolts. And what's plus 20 millivolts plus -70 millivolts? It's minus 50, right? So now, because this summation, I have two excitatory postsynaptic neurotransmissions, and they add up together to not only go to threshold, but exceed it by 5. And now I get an action potential. Okay, because they sum together. If this was, let's increase that for convenience sake. If that was -75 and I only had plus 15, right, then I wouldn't get there. Okay, so that's why summation is really important. All right, so that's spatial. Any questions about that one? Does that one make sense? Okay. Now we have temporal, and temporal is 1. Okay. Let's go ahead and get rid of this one on the bottom. And use this one. This is where frequency comes in handy in explaining things. So one single presynaptic neuron right here fires a +5 millivolt signal to this neuron that's at -70 millivolts. But it does it really fast. If it does it slow, then this depolarization is just going to go into the cell body and it's going to diffuse and it's just going to dissociate and it's not going to do anything, right? But if it does it really fast, it's pretty much like spatial summation where you're getting things almost at the same time. All right. And so you get +5 and maybe it dissociates and then gets down to like +3, but then you get another +5. And so you add your +3 plus your +5. And now you're at +8, but then it gets another +5. And then you see where I'm going, right? Then you get them so close together time-wise that you eventually reach threshold because it depolarizes it so much. Okay, so those are the two differences. The main thing that I would use to memorize those is spatial is multiple presynaptic neurons, whereas temporal is just one. Okay. All right. All right, this is important. If I were you and I had the time, if I didn't have the time, I guess that couldn't be helped. But if I had the time, I would definitely try to recreate this from nothing and be able to write down the numbers and what happens at each number. Okay. I'll just go over it really quickly. I'm not going to do it much into detail unless someone has a question. But what we're looking at here is just the differences or the changes rather, in the amount of ions moving in and moving out, and which ones do which. So that's what I would memorize. So resting membrane potential, right? Depolarization stimulus is at number 2. What would that be? Graded potential, right? Okay, graded potential at number 2. And then the membrane depolarizes to threshold. Okay, so it reaches -55. And then what happens next? Voltage gated sodium channels are open. And then voltage gated calcium channels, or sorry, potassium begin to open slowly as well. And then you have at number 4, the rapid influx of sodium, which is causing the cell to depolarize, get closer to 0. And it goes as far as 30, but I don't know, he's done it a lot of times. How far could it go? About 60, right? Because that is the reversal potential for sodium. Okay. So once it does that, it reaches that plus 30, it I keep doing that because it's a K, I make the K sound. But potassium moves from the cell, okay, to the extracellular fluid in number 6. And what does that do? It doesn't depolarize the cell, it repolarizes the cell, right? Okay. And then at number 7, the potassium channels remain open. And not only is it depolarizing the cell, but it's hyperpolarizing the cell. Okay. So you may ask the question, what's the difference between repolarizing the cell and hyperpolarizing the cell? And that's where we get into relativity, right? We started at -70. Okay. So if I'm at plus 30 and I go down, I'm depolarizing all the way until I get to -70. And if I go any further, now I'm hyperpolarizing because we're looking at it relative to -70. Okay, so that's the difference there. I would know that one because I used to not, and that used to confuse me quite a bit. All right, so number 8, potassium channels close and not as much potassium leaks out of the cell. This is where you're getting into that refractory period. And then number 9, it returns to the resting ion permeability, resting membrane potential, and you're ready for another action potential. Definitely know what happens at each of these numbers. I would for sure be able to draw this and label this for the test if I had the time. Yeah, any questions about that specifically? Sure.
So in the refractory period, going back to not spatial, what is it, roll summation, how does that work? Because it's the same nerve, isn't it?
Yeah, let's go into that with this one right here. Okay, refractory periods. So this is mainly like what's happening with the ion channels and if they're closing or not. Okay, so once you get into this absolute refractory period, if you go above threshold, it doesn't matter if you're getting like synapses that are firing and causing those graded potentials, it's already in an action potential. And so it's already becoming really depolarized. And so extra depolarization isn't going to really matter too much. And you're not going to get like a change in membrane potential that affects it to the point where it fires another action potential because it's already in the process of another one. Does that part make sense?
Yeah.
Okay, so it gets a little bit tricky. And I can't remember exactly what he said he hated about this slide, but I know he said he hated something about this slide. But I'll just pretend like I don't hate it and just explain what it says. But once you get out of that absolute refractory period, then you get into the relative refractory period and another action potential is possible, but it's less likely. If you look right here, this is the excitability. And like I said, it's 0 when your action potential is happening. But after it gets out of that, it starts increasing, but it doesn't get right up next to it, right? It doesn't immediately go back to 100% excitability. It takes a while. And it takes a while because these sodium channels are resetting to their original position. And so are the calcium, or the potassium channels. And once they're set back to their original position, then it's easier for it to, like those graded potentials to cause another action potential. It's been a while since you asked your question. Did that answer it?
Yeah.
Okay, awesome. Yeah, it usually like worries me whenever someone asks a question, the professor goes on like a tangent and I'm like, you didn't answer his question, and now he's like sitting there. So if I ever do that, just let me know. Okay, so that kind of explains that. So thanks for the question. I'll go back a little bit and explain how this actually happens. All right, and we'll use the sodium channel as an example. Okay, so at rest, the activation gate closes the channel, right? And this is the activation gate inside the channel, and the inactivation gate is this little ball outside the channel, or relatively outside. And when the when the activation gate opens, sodium enters the cell, right? There's nothing getting in its way. But during repolarization, when potassium leaves the cell, the two gates reset to their original position, so you don't get any sodium going into the cell and messing up that action potential or that, what's the word? Repolarization. So sodium just leaves because it can't get away or get through this activation gate. And then you get that depolarization stimulus channel, and then it opens it again. But something else happens. The inactivation gate closes the sodium channel. It's entry of sodium stops. Okay. I actually did that in order or out of order. It's supposed to go like this. So yeah, so I would know what happens with the activation gate and inactivation gate as time progresses through an action potential. Right. I think this figure is really cool. I think it helps a lot to understand how that refractory period actually works. So I would know that one for sure, like what happens to these channels as the action potential moves on. Okay, any questions about that one?
Action potential is just at the membrane, right? And it kind of just dissipates.
Yeah, so action potentials go through the axon. And so I don't think you guys covered this very much. I'll do it over here. But each one of these, this is the axon, and then you have like myelin, right? I'm sure you've heard of this term, but you have like the nodes of Ranvier right here. Those are full of sodium channels. And that is the membrane, right? At the axonal membrane right here. And so these, all that sodium is rushing in at the membrane level on those nodes of Ranvier and conducting the action potential. You guys have heard of that, right? Or did I just confuse everyone?
Oh, he was mentioning like they jump, but instead of like on the outside, they jump like from the inside.
Yeah. So yeah, so good question. They do happen on a membrane level. Okay. Oh, we went over that. This. Okay, yeah. So comparing action potentials and graded potential, this is a great summary slide. I wouldn't necessarily memorize this, but I would know this, and it's really counterintuitive. But if you are familiar with this and you're familiar with how that works and this, then that should make sense. Okay. Graded potentials are much different than action potentials, and they have a different purpose. Okay. And if you know that, and you know exactly or somewhat exactly how that works, then you should be good for this slide. This is just a really good reminder or summary slide for that. Any particular questions about this, or why a certain column or certain rows a certain way? Awesome. All right. Okay, so action potentials have certain properties that are really important to their conduction and allow it to actually go. And they're variable based on certain physiological factors that make up diameter of the axon and resistance of the membrane, and if it's myelinated or not, and the frequency. So those are really important for determining whether or not an action potential is actually possible and how easily it will conduct. So this one's really common. Diameter of an axon are a lot faster. And that used to confuse me because I used to try to relate it to water pressure. And if you have an arrow hose and it's really small, then water just like screams out of that thing, right? Just sprays everywhere. But, and this states that the action potential goes a lot faster if the axon is a lot bigger. And I tried to relate the two, like electricity to water, and like the same thing, but they're not. And so it makes more sense if you look at it like this. So here's a really thin axon, and then here's a really thick one. Okay. These walls right here are membranes, but inside the axon, you have proteins, you have stuff in there. And the more stuff you have in there, the harder it is for electricity to actually conduct. Okay, these proteins start to pick up charge, and then the action potential starts to dissipate, and it's harder for the action potential to conduct. So if I have a really small one and I have these really big proteins getting in the way, then it's going to take a while for the action potential to get all the way down the axon. But if these same size proteins are in this bigger one, all right, but I have more space, then the action potential's voltage or current, sorry, is able to go through the axon a lot easier, a lot better. Okay, so that's how I got that straight. Hopefully that helps you. Yeah, so resistance is a really big part of whether or not an action potential is very fast or not. And so you have that one right there, which is axon diameter. That's a factor. But you also have myelin is another factor. Okay, and drawing over there. I left my eraser over there, so I'll just go back. So you also have something else, myelin. And if I have the same diameter axon, but one's myelinated and one's not, I'll go ahead and draw that. This one's not myelinated. This one is, but this one has like myelin around it, right? A little hot dog. Yeah. Then if we're looking at action potential in this one, the current goes through the axon, but this membrane right here is able to like kind of dissipate the charge a little bit. And the charge kind of leaves the axon. All right. But if I have this myelin, sorry, it's a weird way to say it. If I have this myelin, it tries to leave, but it can't. And so it just goes back in because it has this extra insulating layer. It's like if you have like a copper wire and you have that rubber around it, it's harder for the charge to dissipate if you touch it. Okay, so that's pretty much the same thing. So you have diameter affects axon conduction, but you also have myelin. So myelinated axons are a lot faster. Smaller diameter axons are a lot slower. And it goes in a certain order, right? So the fastest axons would be the large myelinated axons, right? And the slowest axon or action potentials through an axon would be this small diameter unmyelinated axons, right? Okay. All right, so I would know what multiple sclerosis is because it's come up a lot for me personally, and knowing what it is has helped me quite a bit prior to not knowing what it is. Every time it came up and I didn't know what it was, I was like, I don't know what that is, and it really sucks. So yeah, I would know what that is. And it's basically just damage to this myelin causing problems with your action potentials. Myelin is extremely important, especially in the CNS, to be able to communicate with your cells in there and to be able to behave properly and have normal function in your arms and periphery, just stuff like that. All right, action potential frequency. There is no difference in the amount of stimulation, like as far as magnitude that affects the action potential. But if I get a bunch of graded potentials at a high frequency, then I'm going to get a lot of action potentials. If I get one big graded potential that depolarizes the cell well past threshold, I'm only going to get one action potential. But if I get a bunch of really small ones that are good enough to reach threshold for the action potential, I'm going to get a bunch because it's always there. So action potential frequency is more important than magnitude, and it represents the stimulus strength a lot better than magnitude does. So I would know that also. You're looking at frequency of the graded potentials and action potentials, not the magnitude. All right. Okay. So synaptic communication, ionotropic versus metabotropic receptors. This is basically just copying what the slide says that I went over. Ionotropics are a lot faster, rapid responses are connected to ion channels. Metabotropic are G protein coupled, a little bit slower, but more widespread. So I would know those properties for sure for synaptic communication. Yeah, any questions about that or what I just went over? I didn't give you guys an opportunity to ask anything. All right, cool. Yeah, so the different types of receptors. Cholinergic would be an example of ionotropic reception, right? Because you have open cation channels immediately upon their stimulation. Okay, and that would be like nicotinic on skeletal muscle or muscarinic. Sorry, that was disgusting. And then adrenergic receptors are G protein coupled receptors, which are examples of metabotropic receptors. And they come in two varieties, alpha and beta. I don't think that that will be on the test exactly what those are, because that seems a little bit we actually go into that a little bit more in depth when we talk about the respiratory system and the cardiovascular system. So that'll be important later, but as of right now, I would just know that there are two types of adrenergic receptors, alpha and beta. All right. This one seems really, really daunting, and that's because it is. I wouldn't memorize this one either. I would just know, I would be familiar with these. I'd be able to list all of these and what class they're in, and what their ligand is. So you have like adrenergic receptors, its ligand is norepinephrine, right? Dopamine receptors, their ligand is obviously dopamine, or histamine receptors have histamine, or GABA receptors, they're like prone to GABAergic ligands. Yeah, I wouldn't really go into how exactly they work with their antagonism and agonism, but I would definitely know if he's familiar with all of those and be able to list them at least. Yes, but yeah, testing over these usually comes up in stuff like drug mechanics or the neural circuitry. All right, I would definitely put this over that for sure, because that gives you more insight on what's important. This is utilizing these and just basically explaining how they work. All right, and we went over this in that lecture, but I'll go over it again. I would also be able to list these out, all right, and know exactly what order those go in. Action potential arrives at the axon terminal right here, right, and causes the whole presynaptic terminal to depolarize. And that depolarization causes voltage-gated calcium channels. Calcium is the second messenger to enter the cell. Okay, and once they enter the cell, this is one of the roles of calcium that I mentioned earlier. You know, they have muscle contraction. They're able to certain factors, but they also signal vesicles to go to the presynaptic terminal membrane and diffuse. All right, so once calcium enters, after it's depolarized and after the gate opens, calcium enters, calcium signals to vesicles, binds to them, and moves them to the membrane. All right, and these docking proteins, an example would be SNARE. I don't know if you're familiar with SNARE, but I wouldn't really worry about it too much. But those docking vesicles release these neurotransmitters packaged in these vesicles into the synaptic cleft. Right, and once they're in there, they diffuse across the synaptic cleft and are able to bind to the postsynaptic terminal or postsynaptic cell receptors and induce a certain response. Okay, so know that in order. Okay, that's really important. That's the basic. That's the foundation of neurobiology, how that actually works. All right. Yeah, I went over this briefly too in the lecture. Neurotransmitter termination. What exactly happens to neurotransmitters that are left in the synaptic cleft? I would just know that they can be returned to their axon terminals for reuse, or it can be enzymatically deactivated, or they can diffuse out. Okay, these are the three main types. They can be returned through, like, I don't know if you're familiar with the NET, and that basically just takes it back to glial cells, or they can be enzymatically deactivated, or they can be diffused. So those three types I would know. An example would be acetylcholine. I would know how acetylcholine is made, basically just made from choline to acetyl-CoA, and then what breaks it down would be acetylcholine esterase. So I would know that enzyme. And so this acetylcholine esterase basically breaks down acetylcholine back into its primitive form of choline. And choline is pumped back into the axon terminal and is then able to be recycled and used further as acetylcholine. Okay, so these are your main types of neurotransmitter termination. This right here is an example of this. Okay. Any questions so far? Okay. Drugs and diseases. Dr. Edwards, I work in his lab. He's really big into drug-induced neurobiology. And so I think there would probably be some questions about this on the test. That's why they put it on here. But different drugs behave differently, and they're all foreign, but they each induce a certain physiological response that's out of the normal. Okay, cocaine blocks reuptake of dopamine, so you have more dopamine in the synaptic cleft that are able to bind to receptors that gets you the feel-good response. Parkinson's disease, the loss of dopamine in the substantia nigra. And they're working on cures for that with drugs that will like help with dopamine breakdown and prevent that. And they also have stem cell therapies used for that. Strychnine is basically just a competitor with glycine, which is an inhibitory neurotransmitter. This is not really a medical drug, it's a poison pesticide. And this is how it works. So if you have a drug that competes with glycine and competes with the natural inhibitory process, then you're going to get a lot of contraction. You're going to get neurotransmission that isn't necessarily supposed to be there. So as much as we talk about excitatory postsynaptic potentials, inhibitory postsynaptic potentials are just as important as excitatory. And you need to have the right amount at the right frequency. And then tetanus also prevents the release of GABA. And so strychnine and tetanus usually kind of work in the same way, but they work on different neurotransmitters. Strychnine works on glycine, whereas tetanus works on preventing the release of GABA. And GABA is also an inhibitory neurotransmitter, but they just do different things. So you can see, even with the common factor being loss of inhibition, you have different molecules that bind to different neurotransmitters. And so you get a lot of variety there too. All right. Divergence and convergence. These, I use their names to remember. Diverge means to spread out. Converge means to come in. And basically what you have to know for those is one presynaptic neuron branches to a large number of postsynaptic neurons. That's divergence. And then convergence is many presynaptic neurons provide input to a lot fewer. So they converge on one part. I would know some examples for this. I don't think they give any. No, they don't. I would know some examples for this. Like divergence would be like the knee reflex. And then convergence would be the reticulate activating system, or the RAS. And that's basically like decision making. Okay. All right. We're almost done with this one. Yeah. Inhibition. Inhibition can also come in two types. And I would know these two types. You got presynaptic inhibition and postsynaptic inhibition. Presynaptic, as the name states, it's in the presynaptic terminal right here. And it basically just stops neurotransmission in that one specific presynaptic terminal onto its specific target. Okay. And then the difference being in postsynaptic inhibition, the inhibition is on the postsynaptic cell right here. Okay. So this inhibitory neuron starts its IPSP, its inhibitory postsynaptic potential, and keeps this neuron from firing completely, which then results in no action potential going down the terminal or the axon, and therefore no response on their target cells. So presynaptic inhibition is a lot more specific, okay, and it's a lot narrower. Whereas postsynaptic inhibition, depending on whether or not this neuron diverges, right? This is an example of divergence. You got one neuron right here diverging into a lot others, okay? Then if that's the case, this postsynaptic inhibition will be a lot more broad scale. Right? So usually postsynaptic inhibition is a lot more widespread than presynaptic. All right, any questions about that? That's a pretty good graphic, I think. All right, so chapter 9, this is the last part. I'm going to go over a couple slides. We went over a couple slides in lecture. So the first part of this would be the development. This one, I don't think you have to know a whole lot, because you can definitely get into this a lot more than just these couple slides. But what I would know is the difference between the neural crest and the neural plate. Right? The neural crest becomes the peripheral nervous system. The neural plate forms the neural tube and becomes the central nervous system. If I remember right, I think he said that he would put that on the test. He had it made and it was fresh in his mind. So I'm pretty sure that will be on the test. I would take his word for it, which is not really that hard to remember. I would just take this into consideration. You have the neural plate right here, becomes the neural tube, but it's called a neural tube because it literally is a tube. And this crest, right, this neural crest becomes a crest on the neural tube, right? Okay. That neural tube becomes the spinal cord eventually. So if that helps you remember CNS, that's also a good way to remember it. All right, this I think he also said might be on the test. The slide after that in the lecture slide said he wouldn't be. So I would remember this one. This is the anterior end of the neural tube. So this part right here of the neural tube becomes this and starts the process of forming the brain. And I would know what section is which. And so you have the most anterior end right here, the forebrain, that eventually becomes the frontal cortex and the cortex itself. And then the midbrain becomes like the pons, and the hindbrains, cerebellum, and then the spinal cord. Right? Any questions about that? I don't feel like you'd really get into detail for that, so I just know the anatomical locations for each one of those. Okay. All right. Gray matter and white matter. This is in the CNS only because we're talking about the brain. And gray matter is just unmyelinated cell nerve bodies. And it's basically just clusters of somas tightly packed together. And I remember it because gray is darker than white usually. And it's gray because they're cell bodies, and the cell bodies contain a lot more stuff. They contain the nucleus, the ER, the Golgi apparatus, all that stuff. Okay, so it appears darker. That's how I remember it. And you also have dendrites and the axon terminals in that. But in the white matter, you just have the myelinated axons, and there's really not much going on there. You have the axon itself and the myelin. And so it's a little bit lighter. There's less stuff. And these right here are basically just your wiring. So all the white matter in your brain is just a bunch of wires innervating certain targets. Okay. And then the blood-brain barrier. Okay. That's important. Also, that's where he left off. And he said that was the last thing that would be on the test. The brain is a very picky thing because it's so important and because it's so complex. It has to be very picky. So you need something to provide the nutrients. You need something to provide the blood and the oxygen, and it has a high metabolic demand. So this has to be well established. All right. And it's established by astrocytes. I would remember that for sure. And basically it just does two things. It provides the nutrients by giving the brain a circulatory system that's not too invasive. And it also protects the brain from toxic substances. Okay, so it does the best of both things right there. This is an image of the blood-brain barrier. It always seemed very abstract to me, so this image really helps. These astrocytes are glial cells that form this by something called foot processes that give you that vein-looking thing. And so I asked the question, well, how does nutrients from the blood actually get into the neurons themselves? So then you look at, all right, well, here's the astrocyte foot. It creates these tight junctions right here in the blood supply. Okay, and those tight junctions bring in the nutrients back into other astrocytes. And these astrocytes are not only innervated on the blood-brain barrier, but they're also innervating certain neurons. And then it's able to just like be the messenger for the nutrients in a sense. Okay. Yeah. So that's pretty self-explanatory. I think that's it. Okay, that's it. Yeah. And the next slide is what he told you not to memorize and be on the next one. Okay. So that's most of what I think is super important. Does anybody want me to go back? Anything in particular? Or have any questions? I'll stick around if you want me to or want to talk to me. These right here. I can. If they're not, just email me if they're not, and I'll go ahead.
We don't know any.
Yeah, this right here, I don't worry about it right now. And everything after it, obviously, don't worry about it. Thank you. Thank you. Yep.